Okay. Okay, so hello everyone. Today I will. Okay, hello everyone. This section will be about gene science and approaches. So the main goal of the main goal of the lecture is to talk in a simple language about techniques and approaches of the one of the most popular direction of genetic manipulation. Today we will talk about what is gene expression and gene science. What is genetic science methods such as knockout and knockdown tools. Also, we will mention about ease of design, ease of workflow, off-target effects, flexibility of application, and their efficiency. So first we will start. So if you have any questions, please interrupt me and ask me. Gene is a framed part of double-stranded DNA or dsDNA that encodes information about gene product. So gene product is a biochemical material, RNA or protein, resulting from gene expression. And each gene product performs some certain function. Gene expression is a process of gene product construction in accordance with the gene code. So here you can see the double-stranded DNA, and as you can see, the double-stranded DNA is made of two sequences with two sequences with some genetic code. So gene expression is going through the central dogma of molecular biology. So what does it mean? Central dogma of molecular biology is the rule that's used for gene expression. And while gene product is expressing, it goes through three forms. The first form of the gene product is double-stranded DNA. So information about gene products is encoded into the double-stranded DNA. So after that, the information from DNA is going to be translated into the RNA. RNA is the second form of the gene product. And in some cases, RNA can be a finished form of gene product, but we will go further, and RNA will be translated into proteins. Proteins is the third form of gene products. And proteins can do some certain functions. For example, they can do gene expression. They can be responsible for traits or features. For example, it can be color of skin, size of your head, etc. Or it can implement some other cell functions. So let's take a look in a picture with more detailed gene expression. So as you can see here, the double-stranded the gene information about gene is encoded into the DNA, and after that is going to be translated into mRNA. DNA is the first form of gene product, mRNA is the second form of gene product. And after obtaining mRNA, it goes out of the nucleus, and information about mRNA will be translated into the polypeptide chain. It is the third form of gene product. So some gene products can be a part of pathogenic processes. For example, some mutated gene products can cause development of cancer diseases or neurodegenerative diseases. So in some cases, gene products can be a part of viruses and bacteria, and these gene products are not mutagenic, but this gene product will support the life cycle of viruses or bacteria. So, and for example, we want to investigate the role of certain gene products, or we want to model some diseases, or even we want to treat them. In this case, we can use gene silencing. Gene silencing is a process of interrupting of target gene expression with the use of gene silencing tools. So let's go back to this picture. So there is two ways how can we affect our, how can we silence our gene. The first way we can affect gene on DNA level. So, wait, I'm sorry. So we can affect or disrupt gene on DNA level, and if gene expression is interrupted on DNA level, there will no mRNA, there will no protein at all. The gene will be totally silenced. The second way we can affect RNA. So if we affect RNA, there will be only protein production. There will be interruption of only protein production. It means that that's not we don't totally turn off the gene, but we will significantly decrease the production of protein that way. So sorry. So the tools that are used for DNA affecting DNA for affecting gene on the level of DNA calls knockout tools. If we affect mRNA, these tools will be called knockdown tools. So it's just an interesting fact about working zone of gene silencing. So if we use, for example, if we use tools for conducting knockout, we have to take into account the gene is a big DNA that has size about 10 in the third power to 10 in the sixth power power of base pairs. So double-stranded DNA is measured in base pairs. And it is illustrated here. So the double-stranded DNA is double-stranded. There are two strands, and therefore we measure it. We evaluate its size with the pairs of bases. So the gene is big, but if we want to work with the gene by knockout tools, we will affect only 10 or 2 or we will affect only little area from 10 to 30 base pairs. It's very low in comparison with the whole gene. And if we talk about chromosome, we cannot even see it. Its chromosome is very big. It's a 10 in the 7th. The size of chromosome is 10 in the 7th, or to 10 in the 9th power of base pairs. It's very big. Chromosome is contains very high concentration of DNA. So, and if we conduct knockout, it will be appearing something there where we cannot see by our eyes. So, if we affect mRNA, so the working so we know that mRNA is single-stranded. It will be measured not in base pairs, but it will be measured in nucleotides. mRNA, so approximately mRNA has a size about 10 in the 3rd power of nucleotides, and the working region of knockdown tools will be about 15 to 14 nucleotides. That is pretty similar to knockout tools, and red mark show us how what region is used for will be affected if we use knockdown tools. But even this little area this little region will be enough for cleavage and destroying all mRNA. So let's talk deeply about knockout and knockdown tools. There are knockout tools with the examples ZFN or thin finger nucleases, Talon, and CRISPR-Cas9. There also is CRISPR-Cas12, I didn't edit. So the general mechanism of knockout tools is described here. So there is our target gene fragment of double-stranded DNA, and there is lockout tools. They specifically recognize double-stranded DNA and just make a double-stranded break. And there are two pathways. That is two pathways, NHHV or non-homologous enjoyment. I will talk deeply about it in the next slides. So in case with this pathway NHHV, here gene will be double-stranded DNA or gene will be disrupted. They will the gene plot that is used for protein synthesis will be disrupted, and there will not be high quality RNA. There will not be high quality of protein, or there will not protein at all. So the second way is HDR or homologous direct recombination. It's a pathway where we use donor DNA. So here we obtain double-stranded DNA break, and we can add some template, donor DNA template that will be inserted into this gap. That in this way we will obtain DNA with a new gene code with new features. Okay, and there is knockdown. Knockdown examples are RNAi or RNA interferences, ISO, DNA enzyme, and ISO is antisense oligonucleotides, DNA enzymes, and CRISPR-Cas13. So there is mechanisms based on use of antisense oligonucleotides, but this mechanism will be this mechanism is similar for every examples. So gene knockdown agents, antisense oligonucleotide, will specifically recognize mRNA and will form ISO mRNA complex with complementary binding here, and nuclease RNase H1 will recognize this nucleus. It will catalytically leave the target DNA. So it means and as soon once we got RNA disrupted, there will no protein. There will no protein. There will no bad functions at all. So let's deeply talk about each example of knockout tools. So the first one is a zinc finger nuclease. So zinc finger is a fusion of two proteins, Focky nuclease and zinc finger domain. So Focky nuclease was isolated from Flowbacterium okeanopolitis, and it's fused or connected with the zinc finger. Zinc finger made of 4 zinc finger domains. Conformation will remind us about the fist with the folded fingers, and so the zinc finger nucleus made of 4 domains, and each domain has this conformation. And this conformation is we obtain this conformation because zinc ions coordinate amino acids, coordinate amino acids, and we obtain this conformation. And these fingers will specifically recognize our DNA, our DNA sequences. So one zinc finger made of 4 domains, and each domain will recognize DNA by triplets, not by one nucleotide, but by triplets. And we have to design our first zinc fingers works only in pairs. We need right zinc finger and left zinc finger to make it work. Why? Because forking nucleases will work only in only as a dimer. So if we have only one forking nuclease, it will not reach. But we need to, and therefore we need to design our zinc finger with the right soft domain and left domain, and they should be designed in such way where forking nucleases will be attached each other in opposite sides of DNA, and there will be DNA cleavage and demonstrated break. So Talon, the next example is Talon, transcription activator-like effector nucleases. It's a kind of analog of zinc finger nucleases because it's also made of, it's also fusion of two proteins, Talon and forking nucleases. And it also required left Talon and left domain and right domain for working. So, but the difference is Talon also made of little domains, and these little domains specifically recognize DNA by each nucleotide instead of triplets as zinc finger. Zinc finger recognize DNA by triplets, and Talon recognize DNA by nucleotides, by each nucleotides. And also Talon is Talon domains will have different orientations from 5' end to 3' end. So in case with zinc finger, we have orientation from 3' end to 5' end. And also Talon is 3 times bigger than ZFM, than zinc finger nucleases, because it has 1000 amino acids residue, but in case with zinc finger, it has only 330 amino acids. So the next one is most hyped and popular Knockout 2, CRISPR-Cas9 and CRISPR-Cas12. So basically, they are about 1,100 or to 1,016 amino acids. They are a little bit bigger than ZFM and Talon, but they key difference from two previous examples is they recognize target DNA not by protein domains, but they recognize them by with guide RNA. It's single or it's two or one RNAs that recognize DNA, and also CRISPR-Cas will require PAM sequences. It's a little marker to that will help for CRISPR-Cas9 or CRISPR-Cas12 to recognize this target DNA as a target, because without PAM they will not reach. And PAM sequence will be strongly required for CRISPR-Cas9 and CRISPR-Cas12. CRISPR-Cas12 is less off-targeted and more controllable. Why? Because CRISPR-Cas12 will require more complicated PAM sequence in the target DNA, and it will cleave DNA with it will left sticky ends. In Russian, it will be liptic acid. In this case, in case with CRISPR-Cas9, there will be blunt ends to nucleic acid. And CRISPR-Cas9 and blunt ends are less controllable than sticky ends. They are less controllable than the sticky ends are less controllable than blunt ends, and I will talk about it later why. So and there is repair pathways. So we obtain double-stranded DNA break. We obtain double-stranded DNA break, and there are two pathways, what I mentioned before. And I will start from homology directed repair (HDR). So as I said before, there will be if we add donor DNA, this donor DNA will be used as a template it will be used as a template for DNA that will fill this gap. And if we use this template, this gap will be filled with new DNA with new gene information. So the second way is non-homologous end joining, NHAG. So and there can be this pathway is occurring if we just don't add donor DNA. So there will be four scenarios. The first one, we will DNA will be prepared and will be prepared to the structure, to the wild type. Wild type means so DNA will be the same as before the double-stranded break. And there is insertion, three other scenarios: insertions, deletions, and frameshift. In this case, the gene code will be shifted, and the information about gene product will be error prone, will be wrong, and the gene product will in some so it depends where we make double-stranded break. On the start of if we make a double-stranded break on the start of gene, they just will know protein, mRNA and protein synthesis. Gene expression will be terminated at all. So in case if we make double-stranded break in the region, in the middle of the gene, there will be just one variant of protein. So I mentioned why sticky ends are more controllable. So with sticky ends, it's more, we will get more chance to knock out our genes. If we have blunt ends with more probability, we will obtain wild type of DNA that will be connected each other, and DNA will be restored, and the gene will still work. But with the sticky ends, it will be more probability. So we will get these three scenarios with the sticky ends. So there will be frame shift or deletion or insertions of DNA, and the gene will be disrupted. So next is knockdown tools, RNA. I saw the enzymes represent. First, we'll talk about RNA. RNA is RNAi is RNA interference. It's usually there are two examples of RNA interference: it's siRNA and miRNA, as you can see here. This is siRNA. Usually it's presented as double-stranded RNA, but whatever, and miRNA. So it's small RNA molecules that have size about 21 to 23 nucleotide duplexes. In comparison with and as an example, the whole RNA has a size about 1,000 nucleotides. It's very big in comparison. This is much bigger than siRNA or miRNA. So usually RNA interference will target one mRNA, and target RNA silencing usually occurs in association with the risk complexes. What does it mean? So we load our siRNA or miRNA into the cell. It will be proceeded with dicer, and we will obtain a little bit shorter version of siRNA, and this siRNA will be associated with the proteins Argonaut and TMDP. It is mentioned here, and this RNA, and we will get RNA protein complex that's called FRISC, and this complex will recognize gene and will specifically cleave it. So next is antisense oligonucleotides, or ISO. They are analog of RNA interferences, but much simpler with the mechanism. And I didn't mention that RNAi doesn't work with bacteria, doesn't work with bacteria because bacteria doesn't have these proteins. So we need to have, if we affect some RNA, we have to have these proteins, and they are presented in eukaryotic cells, but not in the prokaryotic cells. So antisense oligonucleotides is a literal RNA or DNA with size of 20 or 30 nucleotides length. They have much simplified mechanism of work. So ISO complementary bind to target RNA and form duplex RNA and DNA. DNA and RNA. This duplex will be recognized as RNase H1. It's nuclease that catalytically cleave this chromatic complex, and they will be RNA cleavage. So it's another of previously mentioned siRNA. So next is ribozyme. It's little RNA molecule with length of 42 to 50 nucleotides in length, and it catalytically cleave target mRNA without some proteins. It doesn't require some proteins or nucleases associations because this chain can cleave target mRNA itself. But for that, to cleave it, ribozyme will require metal ions such as zinc, calcium, independence of the design. So, and an interesting fact that ribozyme can be inserted into the mRNA or can be a part of RNA. It's mean that mRNA that has ribozymes can cleave itself. But this model usually used for some RNA for research work about RNA work. So this example is mostly used for development of therapeutic agents based on knockdown tools. So go further, there is DNA zymes or deoxyribozymes. It's our lovely approaches what we use in our lab, in our molecular biology lab. It's a short single-stranded DNA with length of 24 to 37 nucleotides in length. They also attach to target mRNA and catalytically cleave it without nucleases or protein, without help of nucleases or proteins, but they also will require metal ions for cleavage. There are two examples of DNA zymes, 1023 and 817. 1023 is more flexible, 817 is more stable. We usually use this variant of deoxyribozyme. Here you can see the picture of how catalytic cleavage occurs with use of deoxyribozymes or DNA zymes. So there is phosphodiester bond of between nucleotides, and if we, for example, we can imagine so DNA zyme or ribozyme attached here, and there will be this catalytic pore, and there will be some metal ions such as calcium, magnesium, etc. And it will promote sort of self-cleavage of RNA. So there will be shifting of what to say. Electronic density will be shifted from this position to this position, and from this position to this position. Sorry, I'm not I'm not good. I'm not a good chemist, but so and there will, in this way, this phosphodiesterase bond will be disattached from this end. It will attach to this end, and there will be self-cleavage for. Therefore, RNA will be self-cleaved. So lineases don't cleave it, but help to RNA be self-cleaved. This is the mechanism. So, and the last one is CRISPR-Cas13. It's a variant of CRISPR-Cas that can recognize target single-stranded RNA and catalytically cleave it. The size of CRISPR-Cas13 is pretty the same, similar to classic Cas9 and a little bit bigger than the talon. So the mechanism is similar. We need a guide RNA that recognize target mRNA. It will also require for PAM, protein-space resistant modifier. So I didn't mention about it before, but okay, we need we require PAM here, but not always. It will depends on where we isolate CRISPR-Cas13 from. Firstly, it was isolated from Leptotrichia stachyi bacteria. So, but we can also use another. Can also use CRISPR-Cas13 from another bacteria, and they will require for PAM or will not require for it. So. This is the picture where we can understand how does knockout, knockdown work. How does it work? And there is the example of mice. So, for example, we want to make our 10 colored mice chocolate color. We conducted gene knockout, and mouse got chocolate color. And as you can see, there is dramatic change in phenotype. If we conduct a knockout, these changes are dramatic. In case we knock down, or if we use RNAi or another tools for knockdown, so the mouse will go from 10 color to milk chocolate color, because knockdown will not cause dramatic change in phenotype. It will be the phenotype will be changed, but not so dramatic as in case with the knockout. So let's talk about ease of design. So first, if we are working with knockout, tools, first of all, we need to choose target gene of choose target site of target gene and verify possible off-target effects. So we can choose our target site of target gene in some genetic databases, but it's very simple. But if we want to verify possible off-target sites, there we can use softwares for zinc finger or talon or CRISPR-Cas9 designs. There is Prognos software where we can check our zinc finger or talon designs for the presence of possible off-target effects. We can just put here sequences of our zinc finger, and this software will show us what's possible off-target. Will be here. So here is the CRISPR scan. And here we can just add target gene, and this software will automatically give us information about what's guide RNA. So I mentioned before that CRISPR-Cas system works with specifically work with target DNA by guide RNA, and there is guide RNAs. And if we put the gene here, the software will give us the possible gRNA with the number of possible targets. So one target of it will be true, and one target will be false. So there is amount of possible targets. So after verifying off-target sites, we need to order or encode proteins or guide RNAs into plasmids. Plasmid is a double-stranded DNA, cycled double-stranded DNA that encode information about protein that we want to deliver into the cell. So we know that that zinc figure or CRISPR-Cas cannot be an eukaryotic cells, but we can deliver zinc figure or CRISPR-Cas into the cells. It's hard to deliver protein, a full protein into the cell, and the variant of decision, the problem is use of double-stranded DNA that encode this protein itself. So once we deliver this plasmid that encode information about challenge CRISPR-Cas into the cell, it will be expressed. So here we and this way we order we decide to order or encode proteins or single guide RNA into the plasmids. So as you can see, we need to use two plasmids for zinc fingers or talents because, as I said before, zinc finger and talents will work only in pairs. So here will be left ZFM or talent, and there will be right ZFM and talent. So in case with the CRISPR-Cas system, we will also have two plasmids. The one will encode guide RNA, single guide RNA, a lot of names, and CRISPR-Cas, and the second plasmid will encode CRISPR-Cas9 or CRISPR-Cas2 protein. So, and okay, so in case with zinc fingers, as I said before, this zinc finger will be encoded, or talent will be encoded in this plasmid, and this zinc finger of talent will be encoded here. So, but there are disadvantages about zinc finger and talent. About design with design of zinc finger and talents. So encoding of whole proteins into the plasmid is very step by step, and it's kind of time consuming. It will take about 1 or 2 weeks of synthesis, and new DNA target will require new ZFM or talent, new pairs of ZFM or talents. In case with CRISPR-Cas and CRISPR-Cas9 and CRISPR-Cas12, they will require for only new guide RNA. So we don't need there is no need to make new protein for new target, as in case with zinc fingers or talents. We need to just remake single guide. We need to remake guide RNA. So that is the profit of use CRISPR-Cas9 or CRISPR-Cas12 systems. So ease of design of knockout agents. So firstly we need to choose target mRNA sequence, target gene. For example, there is a target for DNA enzymes. There is a target for CRISPR-Cas13. And after that, we need to verify knockdown agents for possible off targets. So, as in case with the knockout tools. So we can use the software that's called GeneScript. And here we can verify our designs of knockdown tools for possible for possible off targets. So we inserted here the sequence of our knockdown tools, not gene, or knockdown tools, or siren, antisocial nucleotides, and enzymes. And for this sequence, there will be this software will give us information about possible targets that will be affected with this design. So in this case, there will be about 8. Yes, there will be 8 targets. So go further. So we verified the damages. And here is encoded. We have 2 ways. We can encode our knockdown tools into a plasmid, or we can use itself. We can use nature form or native form of gene of tools for gene knockdown. So honestly, I would prefer use to this one because because delivery for native knockdown tools are more controllable than plasmid. So if we insert our plasmid into the cell, and it will produce a lot of knockdown tools, it's not so controllable. It's less controllable. It's hard to control. It's better to use this. It's better to use deliver only native form of our knockdown tools. So and advantages of knockdown design is very easy to encode in comparison with knockout tools. We don't need to encode whole proteins. We don't need to encode a big amount of DNA. We can encode information about little RNA or antisense oligonucleotide. It's that make our work much easier. And as a consequence, oligonucleotide synthesis may take about 1 to 5 days. So the workflow of how can we implement experiments with our tools. So in case with knockout tools, okay, so we got our knockout tools, talents or CRISPR-Cas. We got the plasmids, and these plasmids we transferred into our cells. And okay, 24 or 22 or 48 hours. After 24 or 48 hours, we need to check what we got, so we can use two types of checking the result of successful knockout. We can use standard sequencing, or we can read DNA, or we can do a Western blot. So in case with standard sequencing is the cheapest, one of the cheapest type of sequencing. We can just obtain the DNA chain of cell which has knockout. Or we can do Western blot. This is the shell, and inside the shell we can load the target protein. And if we, for example, we load target protein, and there is a picture. If the protein will be presented into the cell, if the target protein will be presented into the cell, it will give us light here. But if target protein is knocked out, so there will be no any bands here. And there is workflow about knockdown tools. So there is the similar scheme as was represented before. So we got our knockdown tools. We loaded up. We got a plasmid of it. or we got the native form of knockdown tools, we load it into the cells, and after that we need to check the results after some time. So we can also use the Western blot analysis, or we can use a quick PCR, quantitative real-time PCR. So in case with the real-time PCR, there we load our DNA template into from the cell, which we conducted knockdown with, and here and from this cell we will load DNA templates and into this apparatus, and this apparatus will show us the graph. So this is the graph, and this is the threshold. So to understand was knockdown successful or not, there is CT value. If gene isn't knocked down, this CT value will be obtained on the left side of the graph, and it will mean it's okay. The gene is working, it's okay, and it didn't knock down. If knockdown was successful, so this CT value will be on the very right part of the graph. And Western blot is so as I mentioned before, there is a gel, and we load here the target protein. And as I said before, that if we have protein, it will give light. But if we have protein with a decreased amount, it will give barely noticeable, barely visible band here. It looks like there is no band, but trust me, there is some band. It will represent some little barely lighting band. So if you got tired as me, we can make a 10-minute break if you want. Okay. If you have some questions, you can ask about.
Yes, slide number 10.
Okay, 10 minutes for.
It's the region, you mean the region B here, it's on the body ATM, or it's different to be in the coding sequence?
Yes, you're right. It's better to mark this region on the coding sequence, but I mark it here just to imagine how the size, the significant difference between size of mRNA and size of region where knockout tools are working.
Just the size, not the place.
Sorry?
Just to illustrate the size, compare the size, not detect the place.
Not to get the place.
The region.
Sorry, I didn't catch this. Okay, I hope you got. I hope you got what I mean.
She's trying to say that it's what you've illustrated, meaning that you are only trying to illustrate the size.
Yes, I wanted to only illustrate the size. Yes, I wanted to only illustrate only the size of the region where knockdown tools is working.
Is it possible to determine which of them, non-religious combination or molecule-directed, is it possible to determine, to pick which one you want?
You mean what's to onto you?
Yeah. You have like non-monogamic combinations, right?
Yes.
And then all the directed combination.
Yeah.
Is it possible to determine like okay, I want monogamic combination, not
Once HDR is not a condition.
yeah, is it possible or I will just use this chance?
It's you mean that is it possible?
Is it possible to choose like okay, I want this?
Yes, it's possible to choose because if you want the password with HDR, you mean don't use donor recommendation. It will be enough to just so you can show you. So in this case, we can add we just need to add donor template. So okay, understood. Okay. So we broke our DNA and we in ourselves and we can transfect donor DNA. And this donor DNA would be a template for filling this gap. So if we want this pathway, we can just avoid adding HDR. Wait, sorry. We can just avoid adding donor DNA. Sorry. No, you can see also if you want to hear more. If you want to hear more, I don't think you need to hear more students.
No, it's okay. Is that? That's right. 16 as well. I keep laughing at them. Oh, my phone, because it's over there. The shooting is Friday,
Okay, let's continue. We'll not wait for others. Okay, so if we talk about let's talk about off-target effects. So talk about knockdown tools. There will be usually with non-target interaction with the DNA, because and they can cause irreparable DNA repair, especially if the use of CRISPR-Cas9 system. So what does it mean? So there is double-stranded DNA, and zinc finger or talon or CRISPR-Cas off-target bind with DNA DNA, and this DNA is strong. It's not DNA, not target DNA. And in this case, it might be we might cause growth of DNA in the we can break DNA and in off-target sites. It can contribute to damaging of chromosomes. It can cause deletions or insertions or translocations. It can cause very significant exchanges inside the chromosomal cells, and can cause some bad diseases such as cancer. So we need to be very careful with with the chromosomes this way. And the second off-target effects is immunotoxicity. Usually it depends on which delivery agent we use, what we use for delivery. So we can, if we use, if we talk about immunotoxicity, the main rule, it will depend on what we use for delivery. So if we use adenoviruses delivery, it will cause immunogenicity. It will cause some immune response. In the same way we have with another chemical reagents. For example, there are also a lot of chemical carriers that can cause not specific cytotoxic effect. And also plasmid that's usually used for delivery knockout tools may cause cytotoxicity, especially in embryonic stem cells. So so if we want to solve off-target effects with knockout tools, we can, as I said before, we can use special softwares for preventing, for predicting off-targets. As I said, I was talking about prognos, gene script, and there is also gene design, Cas design. Cas design is one more software that can predict possible off-targets of CRISPR-Cas system that you use. For example, you have some target gene, as I said before, with the CRISPR scan. So you insert the whole sequence of target gene, and it will give us information about possible guide RNA which we should use, and possible off targets that can be affected with the designed CRISPR-Cas system. So the one of the solution of off-target effects with non-target interaction with DNA is use of bioinformatical softwares. So the second way, if we work with the CRISPR-Cas systems, we can make it more complicated in terms of various CRISPR-Cas nucleases. And what does it mean? So usually we got used to use only one CRISPR-Cas system that attached to DNA and ROC. But in case with CRISPR-Cas nucleases, it's a modification of CRISPR-Cas systems that can cleave only one DNA. And if only one DNA is cleaved, there will no double-stranded break, and this break of single-stranded DNA will be easy repair. But if we use two nucleases, they will cleave DNA here and here, and there will be double-stranded break. And what is the profit of use of CRISPR-Cas nucleases? Nucleases are more. They require two DNA chains for recognition, like the thin fingers or thousands. So ordinarily CRISPR-Cas will require only one DNA, but nucleases will require for two DNA that will significantly decrease the off-target work of CRISPR-Cas system. So ordinarily CRISPR-Cas can just recognize this sequence, and despite its off-target, it just can attach to it and clear it. But with use of two CRISPR-Cas systems, we will decrease the possibility of off-target cleavage of DNA. And here you can see that this approach will reduce off-targets 50 to 1,500 fold. So, and if we want to solve the problem with the immunotoxicity, we can use native form of proteins. So there is thin figure that protein that can be directly delivered into the cell, and there is CRISPR-Cas ribonucleotide protein form that's without plasmid, is directly delivered into the cell. So it will help avoid some undesirable cytotoxic effect from plasmids or from some carriers like viral carriers or another chemical carriers that can cause immunotoxicity, undesirable immunotoxicity. So and off-target effects are not awful for if we use knockout tools in work with cell. What does it mean? So if we work with cell, we can modify. We have a culture of cells, and we can modify them. And we need only one cell from all the petri dish with successfully knocked out, which is successfully knocked out, and we can use one cell and breed it in another petri dish. So if we work with, and if we have other cells with undesirable knockout, with the undesirable effects after knockout, we can just waste it and use only one cell to breed it, and we can work with successful knockout cell later. but if we talk about delivery inside into patient with viral vector or non-viral delivery, so these off-target effects of knockout tools will be significant, will be very significant. They will cause chromosome translocations, some damage of DNA. It will be not controllable, and it will be very dangerous to use it on a human. And therefore, CRISPR-Cas and in vivo balance are still in a study of development of less of targeted form of their less targeted form. So if we talk about knockdown, there are also a problem of non-target mRNA binding. So ISO or knockdown agents can bind to ROM mRNA as knockout tools, and that means that ROM mRNA or ROM gene will be silenced. And there is also a problem of immunotoxicity. So if double-stranded RNA is longer than 30 base pairs, it can increase off-target effect and can cause non-specific toxicity via activating interferon pathway. But in case we, and here, if we use antisense oligonucleotides with wrong chemical modifications, they can also cause pro-inflammatory activity. So to avoid non-target binding, so we can use also bioinformatician software. It's PFR Threat 1.0. Here we can upload our information about gene, what we want to silence. So this is a database where we can insert the gene, and there will be ID of gene. In this ID, we can insert to the PFF 1.0, and this software will give us information about, will give us variants of antisense nucleotides that can be used for successful knockdown without minimal off-target effects. So also we can use sugar or we can use chemical modifications that can decrease pro-inflammatory properties. So there's an example. So they can be attached to the chemical structure of our knockdown tools. And we are close to the finish. There is flexibility. So if we talk about flexibility or versatility, the amount of ways where we can use knockout tools. So zinc finger balance can be used as for knockout or knock-in. Knock-in is a technique where we use our donor DNA chain that will be inserted into the gap. So into the gap after double-stranded break. And zinc finger and talon can also be used as gene effectors. What does it mean? So we have zinc finger or talons, and they can be modified. We can delete 40 nuclease and add a factor that can, and this information will, this modification will bind to target DNA and will launch the expression of gene. So CRISPR-Cas also can be used for knockout and knock-in, and can be used also for knockdown. There's CRISPR-Cas 13, it was mentioned before. can be used for base editing. So we can modify our CRISPR-Cas9 with a special enzyme that will change the genetic code of target DNA. Also, CRISPR-Cas may be used for gene repression or gene activation. So in case of gene repression, CRISPR-Cas will be modified with the protein which will repress the gene expression. And in case with the gene activation, Cas9 will have modifications that will launch gene expression. So we can control gene expression with the use of CRISPR-Cas system. If we talk about flexibility of knockdown tools, there are three. Usually we can use them only for cleavage of RNA. So it will be if we use siRNA, mRNA, or multicyclic we can use them only for RNA cleavage and nothing more. CRISPR-Cas13, it's a variant of CRISPR-Cas12, can be used as for knockdown or for can be used for RNA editing. So we can modify our target mRNA with new nucleotides, or we can use CRISPR-Cas13 for detection of mRNA. So CRISPR-Cas can be modified with fluorescent protein that will give light for when CRISPR-Cas will detect our target mRNA. That will help us. For example, this modification can help us to detect the track how RNA is traveling inside the cell. So next is DNA enzymes or ribozymes. Ribozymes and DNA enzymes can be used for catalytic RNA cleavage or can be used as biosensors. What does it mean? So this is DNA enzyme based probe that recognize RNA, and if it recognizes RNA, it will have catalytic form, and this catalytic form will cleave signal molecule. Signal molecule can be cleaved only in the presence of RNA. Without RNA, there will be no cleavage of signal molecule. So in case with ribozymes, there will be ribozyme with an optimal addition here, and this ribozyme will detect the target molecule. It can be some DNA or some protein, and if this protein attached to this space, to this part of ribozyme, it will be self-cleaved in this site, and this RNA will be transformed into this information that will give light in the combination with the fluorophore. This fluorophore will be catched with this RNA structure, and it will give us light. So, and efficiency. So I just said it can be a little bit, not a little bit, it can be subjective, but because it's very hard to really assess the efficiency of any knockout tools and compare them each other. So there is a template where there is each tools what we were talking about before, and with some literature, I managed to find out about some information where usually CRISPR-Cas and RNAi has similar on-target efficiencies, and I mark them as 5. So if we talk about zinc finger or talent, so talent has lower efficiency than CRISPR-Cas system, but there is no certain information what is better, talent or ZFM. There is no information about comparing their efficiency. But I think that if ZFM and the talent has similar structure, similar mechanism of use, it can be that it may be that talent and ZFM have similar efficiency. So I decided to mark ZFM and talent as 4. And if we talk about antisense oligonucleotides, ribosomes and DNA enzymes, so here we can see that from this, it's gathered from the paper, where siRNA was mentioned as the best tool for knockdown. So more active and more active than ISO, ribosomes, and DNA enzymes. So then I decided to put ISO and DNA enzymes less than RNAi. ISO will mark as 4. And ribosomes and DNA enzymes. So it's a very arguable moment because they have varying efficiency because they are highly dependent on the presence of metal ions inside cells. So there is the article that describes this moment about DNA enzymes and ribozymes. They generally exhibit little or no catalysis at the free divalent metal ions. Divalent metal ion concentration inside the cells is neat, so not always cell will have some concentration of metal ions that is required for effective cleavage of RNA with the ribozymes and DNA enzymes. So that I decided to mark from there is 2 to 5, but it will be more it would be fair if you mark them as from 0 to 5. And little facts, there is talent, again, very high specificity, but zinc finger nucleases and CRISPR-Cas can be upgraded and they can be delivered as proteins, and protein forms of ZFM of CRISPR-Cas will work much faster than if we deliver them in the plasmid form. So if we deliver plasmid form, plasmids of CRISPR-Cas or zinc fingers, we need to wait for expression of these proteins inside the cells. But if we deliver proteins to the cells right now, they will work right now. They will be more effective. Okay. So CRISPR-Cas13 also can be upgraded like CRISPR-Cas12, CRISPR-Cas9, RNAi or siRNA and mRNA can be delivered as plasmid form. Double-stranded DNA, and as I mentioned before, ribozymes and DNA enzymes can be dependent on the presence of metal ions. So, and there is total comparison of general efficiency. So, what will we use for? What would we like to use if we want to make an experiment with, for example, we want to work with cell, we want to use the most effective tools on these collections. So the tools were assessed by Marx for the next moments. It was efficiency, ease of design, ease of workflow, of artifacts, flexibility, and availability. And there is total mark that is assumed for every moment of each knockout and knockdown tools. And we obtain, and I think that it's my subjective meaning, my subjective position, but These values were obtained from some articles or from some previous slides, but I got that CRISPR-Cas13, ISO, and many enzymes have the highest value of the highest value 25, the highest mark, but the unpredictable action of the enzymes it's a big dot for us, and so that I think it would we are working with DNA enzymes, but we have a problem that they don't work in cells, so that we think so that from these three variants, this variant is cannot be suitable for our research, so that we now we are using antisense nucleotides because they really have high level of efficiency inside the cells, and they're also they're also the CRISPR-Cas13, but we don't use them. We still don't use them, but I think once we will afford ourselves to use it. So this is general efficiency if you want to work with cell, and general efficiency if we want to work with therapy. If it's therapy, there is some situations we would like to use antisense oligonucleotides and CRISPR-Cas systems, and there is a little difference. In case we work with cell, off-target effects of knockout tools are low or not significant. But if we talk about therapy or delivery delivery resistance inside humans, there will cause unpredictable off-target effects. That's very difficult to control. So this is the best tools what we can use now, and thank you for your attention.